Good day, this is the Professor, CBMD PhD, and today's Monday, April 24th, 2023. It is 7:15 and 0 seconds, AM Eastern Daylight Time, and today's research, cell mapping. So let's hop to it here. Researchers reveal a map to study novel form of cell-to-cell communication. Date April 20th, 2023. Source: Baylor College of Medicine. An international team of researchers lays a foundation to examine how extracellular RNA and its carrier proteins found in bodily fluids function in a healthy as well as a disease setting, potentially providing a means to accurately implement early detection and monitor disease processes. Ribonucleic acid or RNA is one type of genetic material that is present inside all living cells. It is mostly known to act as a messenger carrying instructions encoded in the DNA for synthesis of proteins. In recent years, research has shown that RNA not only exists inside cells, but also exported from cells as extracellular RNA and plays a role in cell-to-cell communication. Extracellular RNAs exist in bodily fluids outside of cells, where they can associate with a variety of carriers, including RNA-binding proteins, but the cargo and distribution of RNA-binding proteins across biofluids is largely unknown. The goal in this study was to fill that gap, this major gap in knowledge of the understanding of the role of RNA-binding proteins as carriers of extracellular RNA in human bodily fluids. The findings open a new road toward understanding extracellular RNA biology and provide new opportunities for the development of extracellular RNA binding protein and extracellular RNA liquid biopsy biomarkers. The researchers applied computational analysis to identify extracellular RNA binding proteins in plasma, serum, saliva, urine, and cerebral spinal fluid. The computational predictions were validated experimentally at about 80% in both plasma and cell cultures in the laboratory, suggesting high specificity for the computational method. And with this information, they developed a map of candidate extracellular RNA binding proteins and their extracellular RNA cargo in bodily fluids, expanding a landscape of potential biomarkers that can now be studied in liquid biopsies and used to track normal and disease processes. And this is a professor of microbiology, bioinformatics, pathophysiology, and leader of research. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.